Hi 107 fans. Today we're going to talk a little bit about chromosomes and alleles and all that other fun stuff. So let's review. Remember, if you're a cell, and let's say we're 2n equals 6, how many chromosomes are we going to have? 6 chromosomes is what we should have if we're 2n equals 6. That means we have 3 homologous pairs, right? Just like the same stuff that I've been talking about in class, this pair, right, would be chromosome number 1. This pair would be chromosome number 2. And this pair would be chromosome number 3. Okay, now remember, we're drawing them as sticks because that's the only way we can see them, but they're actually in their spaghetti shape until we decide to go through DNA replication. Okay, now we're going to look at just one of these pairs of chromosomes so we can actually look at alleles and talk about more about what that actually means. So if we go back to our cell and we ignore the rest of the chromosomes, and we just look at one homologous pair. Remember, if this is chromosome 1, it's got all of the same genes on both of these chromosomes, right? This is chromosome 1, and this is chromosome 1. One of these chromosomes you got from the sperm that made you, the other chromosome we got from the egg that made you. Okay, so let's pretend like the blue line is the chromosome from the sperm that made you, okay? And let's talk about earlobe shape because that's a nice, easy example. It's a one-gene trait in human beings, and we can follow the genetics of that. And so let's say you have the detached earlobes, the floppy kinds. Those are dominant. And let's say you got the dominant allele, we'll call it capital D, on the chromosome that you got from your dad. And let's say you got the recessive allele, little d, from the chromosome that you got from your mom. Okay, so if this is your genotype, go back and write your genotype here. Your genotype is big D, little d, heterozygous. We know that detached for big D is dominant because we told you that. So what would your phenotype be if you were big D, little d? Why, yes, you would have the detached earlobes. Floppy. Okay, does that make sense? All right, let's go on and look at a cell, and let's look at 2 of your chromosomes. Okay? Because that will get into the 2-gene trait, which is what we're going to talk a little bit more about. So again, let's say here's your genotype for the earlobe shape, and let's also then look at hitchhiker's thumb. Okay, hitchhiker's thumb is going to be on a different chromosome. So hitchhiker's thumb, again, we'll say you've got hitchhiker's thumb allele from mom and the recessive allele from dad. So you're heterozygous for hitchhiker's thumb. You're heterozygous also for detached earlobes. Now, if this is your genotype, again, if this is your genotype, big D, little d, big H, little h, right? You're a heterozygote at both alleles, at both genes for both alleles. Okay. So what are all the possible gametes that this individual could make? And again, we're just going to pretend like they have 4 chromosomes. We're going to ignore everything else. So what are all the possible combinations? Well, we could have during meiosis, right? First we replicate the chromosomes. Okay, we're not going to... All right, let's replicate our chromosomes just so we don't get all crazy. Right? The sisters are going to have the exact same alleles. Oops, can't draw that D. Because they're exactly the same by DNA replication. And now, in meiosis 1, the homologous chromosomes come apart. Right? So we can imagine if they were lined up like this, both the blue guys would go that way, and both the red guys would go in the opposite direction. And what we'd end up with would be, we'll draw them one over here and one over there. Right? We would have haploid cells at the end of meiosis 1. Right? This one, I'm only going to put the big D on one side because it's too small. Well, I'll try. Oh, there, I did it. And the big H and the little D's. Okay? And then, of course, once we go ahead, and go back, I'm going to go over here, and we make the actual gametes. That's big D, little h, big D, little h, and this is big H, little d, big H, little d. Okay, so that's one possible scenario of how the gametes could get together. Big D, little h, or big H, little d. Now, if we remember what happens during meiosis, it's random alignment at the metaphase plate. So what if, instead of traveling this way, this guy decides to go this way? This guy goes that way. And the bottom guys do the same thing that they were doing before. Well, we draw another whole set of gametes, right? Now we'd have the little d guy going with the little h guy, and that would be the end of meiosis 1, or big D and big H, the end of meiosis 1. So if we think about all the possible ways these could combine together, right, and if we had big D, little d, big H, little h, right, we can have the DH come together, we can have the little d, little h come together, we could have the big D, little h come together, or the little d and the big H come together. 4 different types of gametes that can be made from this heterozygote. So if we go back to genetics and we look at the cross, and each one of those can make all of those different gametes, right? This guy can do the exact same thing as his partner. This is where we end up with the square that has 16 spaces. Does that make sense? So if we wanted to actually fill out these, we could. It would take us a long time, but we could. And what we would end up finding in this dihybrid cross of 2 heterozygotes would be a 9:3 to 3:1 ratio of the offspring. Again, this has been a little tutorial about chromosomes, alleles, independent assortment, which creates the different types of gametes, and then how we would put them together in a Punnett square. I hope this was helpful. Thanks, guys.